Hi everyone. I finished up last time talking about some of the causes of cancer that we know about. So things like inherited mutations, certain viruses can cause cancer, radiation, sometimes immunodeficiencies, and certainly chemicals. And one thing I wanted to point out too is that particularly things like viruses and immunodeficiency, these are a lot about interactions, and we don't always know how much. Uh, Viruses can really cause cancer, but also the interactions between viruses and bacterial infections or predispositions genetically that might make someone more susceptible to cancer. Okay. So I think we're learning a whole lot more now about the immune system and its role in cancer, and then how viruses and immune deficiencies might affect cancer rates as well. Okay. So what we do know about cancer is that cancers develop gradually. Okay. And so it's not usually that one gene mutation is going to cause cancer. Okay. So it's actually a multi-step model of cancer development where, let's take colon cancer for an example. Okay. So here you have normal colon epithelial cells. And a single gene mutation, like the loss of a tumor suppressor gene like APC or any tumor suppressor gene, might cause there to be a small benign growth. Okay. And so someone might be a polyp former, meaning they continue to have these small benign growths, but they're benign, right? They're not going to go past the borders. They're not going to be very dangerous. Um, as doctors are going to screen for these things, they might remove the polyps, but overall, it's not going to be a malignant cancer yet. Okay, so just a single change in a gene can result in small growths, benign growths. But then, if there's a second or third mutation, maybe you have activation of something like a RAS oncogene or loss of another tumor suppressor gene, then through those mutations in those three genes, now you get a larger benign growth like an adenoma. Okay. And then additional mutations will get you to a malignant place. So it's not that you have a single mutation cancer, that you have multiple accumulating mutations that over time allow for tumor formation and eventually metastasis. Okay. And what we know is that, and with some evidence for this, is that alterations in several cell cycle genes, when they accumulate, can cause this to happen. And this might explain how some families are predisposed to certain kinds of cancers genetically, because if they already have um, maybe one mutation in a cell cycle gene, adding another one in somebody, or a chemical change in the environment that causes another mutation, now you might have more likely to have cancer. Okay. The other thing we know, in addition to this being a multi step model of cancer development, is that DNA repair is a big deal. So typically in cells, there's DNA repair enzymes that are going to fix mutations so they slow this process down. So most of our cells, most of the time, you're going to see DNA repair enzymes fixing the broken DNA, causing apoptosis in cells that should die if they are mutated. And so what we actually find is that most cancers involve malfunctioning DNA repair mechanisms, because normally you get a mutation, DNA repair enzymes fix it. But if you have a mutated DNA repair mechanism, now you can't repair the mutations that do happen. And so you get accumulation of mutations and then eventually malignant cancers. Okay. So since mutations in genes are responsible for finding and fixing this damaged DNA, cancer cells often show a lot of regularities in their genome. Okay. So if we look at this is called a karyotype, right? Where you have, you're laying out everybody's chromosomes. And in a normal cell, it would look like this, where you have all 23 pairs of chromosomes laid out, and these look normal. If you genotype and do the karyotype of a cancer cell, it can look like this. And what you see is you see some chromosomes gone entirely, some extra chromosomes, right? Bits of chromosomes on other pieces of chromosomes. And this is all because you are getting, there's problems with DNA damage, right? And mutation. So normally all these things, if this was happening in a dividing cell, would be fixed or the cell would die. But cancerous cells are able to continue to keep this and keep dividing because they're not paying attention to those normal DNA repair or those normal cell cycle control mechanisms. So that's how you can tell. when they do a biopsy on somebody, they'll do a biopsy on a piece of tissue and they can look at the karyotype. And if it looks really damaged like this with lots of extra DNA or extra bits of DNA or small pieces of DNA, it can be indicative of cancer, and they can start looking for things like the mutation basis or the genetic basis of that cancer. Okay, so one example that we've learned a lot about recently has been these BRCA genes, so BRCA1 and BRCA2. And BRCA1 and BRCA2 are tumor suppressor genes. So everybody has them. Everybody has a BRCA1 and BRCA2 gene. And the proteins that are made from these genes typically are ones that recognize DNA damage and initiate DNA repair. Okay. What they found is that 10% of women that develop breast cancer, okay, and 60% of women with the mutation in the genes for BRCA1 or BRCA2 develop breast cancer. So I want to point out that, again, everybody has a BRCA1 and a BRCA2. It's the mutation and a particular mutation in BRCA1 or BRCA2 that can confer this breast cancer risk. Okay. And BRCA1 or 2 mutations are also risk factors for ovarian cancers and some other kinds of cancer as well. So typically, general population, about 10% of women will develop breast cancer. 60% of women with BRCA1 or BRCA2 mutations are likely to develop breast cancer. Okay, and so a couple of years ago, this became in the media more when Angelina Jolie was tested for BRCA1 mutations. So her family has had a history of breast and ovarian cancer, and so she was tested for BRCA mutations. Okay, and there was a mutation found in her BRCA1 gene, and the conclusion from the genetic doctors was that she had an 87% chance of developing breast cancer and a 50% chance of developing ovarian cancer, essentially because the mutation in that BRCA1 gene is not going to allow fast enough repair of the DNA damage. And so you're going to have really fast accumulations then of mutations in her cells that could cause cancer. And so recommended by the doctors, and she sort of famously had her breast tissue, ovaries, and fallopian tubes removed to prevent cancer development in those tissues. Okay, so it's tissue specific, which is pretty interesting. And this was part of the movement, but has led to a lot of other folks too thinking about BRCA1 mutations and maybe being tested for them. And so when this happened, Myriad Genetics, so a company out of Utah and the University of Utah, discovered mutations in these BRCA1 and BRCA2 genes that increase cancer risk. And so what they attempted to do was at the time, they were able to patent naturally occurring mutations so that only they could perform the genetic tests. So if you were going to get a BRCA1 or a BRCA2 test to see if you had a mutated gene, it would be $16,000 per test. You could only get it from this Myriad Genetics company and the University of Utah, and you couldn't get it from any other biotech company. Okay. What happened, though, then was that in 2013, the Supreme Court ruled that you can't patent naturally occurring genes. Because it wasn't that they were patenting technology. They weren't patenting something that they had developed, right? They were patenting the gene, which now Supreme Court said you can't do. So now, and that's really changed, right? So typically, policy is going to be a lot slower than science. And we're seeing this with CRISPR, right? But now the Supreme Court ruling was really important because nothing naturally occurring, so no naturally occurring genes and mutations can be patented, which means that companies can't keep the sequence from the public. We're seeing this now with the COVID-19 virus, right? Somebody couldn't patent the virus genome, which means that multiple companies and multiple healthcare institutions can work on it and provide more care, right? So now BRCA1/2 testing is around $3,000 or less, and it's covered by most insurances. The Affordable Care Act stated that insurance companies must cover it if it's been recommended by a physician. So what I say for folks is that if you have family members who've developed breast or ovarian cancer, especially early on, before the age of 50, I encourage you to talk to your doctors about BRCA1/2 testing because you might be a good candidate for testing for that, and then eventually maybe some preventative measures, right? So it'd be good to know about cancer risk ahead of time if possible. Yeah. Okay. And so I put this graph always, or this figure at the end of my cancer lecture, not just to have you memorize anything, but just to show you that, whenever people talk about like, oh, let's go ahead and cure cancer, like, why aren't people trying to cure cancer? Well, the thing is that a lot of people are trying to cure cancer. And two is that it's really, really complicated, right? So all of these little figures here, each one is a different gene that we know is related to cancer risk and different kinds of cancer risk, okay? So what you see is you see groups of genes, right? There's a whole bunch of genes that make cells evade apoptosis, meaning that they don't respond to cells telling them to die. We see a bunch of genes related to proliferation, so cell division. We see a bunch of cell cycle genes here, chemotherapy resistant genes, repair, failure mechanisms, genomic damage. So hundreds to thousands of genes that we know are involved in cancer risk. And so it gets really complicated, right, in terms of how to actually cure cancer or prevent it in the first place. Okay. And so, as usual, you guys are going to have the practice problems. But what I want you to do also is to comment on the discussion board. So your second assignment for this class is going to be to comment on the discussion board, and you must post an original comment or question about cancer and then also respond to two of your classmates' comments. So I want to get a discussion moving on the discussion board. I will comment on there as well and try to answer some of the questions. But I want you guys to think about this and think about what we know about cancer, what you want to know more about cancer, and sort of where we are in our knowledge about cancer. All right. I will see you soon.